Hello everybody, and greetings from the Museum König, where I spent the last weeks digitizing tape recordings of soundscapes, mainly from Kinabalu National Park, Sabah, Borneo, with a focus on frogs, cicadas and Orthoptera, which are the main focus of my talk. From song libraries to corpora, a case study on extracting songs from environmental recordings. So please apologize that I couldn't make it in person, but I prepared this video, which will be available on YouTube, open for comments, as a compensation that I am not available for questions and suggestions here. Also, you find links and comments in the comment section of the YouTube video, and of course you're welcomed to add your own comments and suggestions, because you will see that what I propose here is sort of open-ended. So these are the animals I'm talking about. Orthoptera, this is a katydid, and all the specimens in the museum collection which have recordings from my side are labeled with a green label. And you see here is the number of the cassette, the audio cassette, and there you can find recording. So the challenge was to digitize the old cassette recordings and to bring specimens and recordings together. So these are the specimens, for example crickets, in this case from South America, and these are the typical cricket songs, usually at a pure carrier frequency with a certain pulse rate and a definite frequency. So here you see specimens and songs belong together. Later on, once we have made this connection, we can upload the files to the Orthoptera Species File, which has the big advantage of using the latest taxonomy of Orthoptera, which of course is in change, and there are a lot of species to be described. So, now let's go from the lab to the field. These are some sites where I made major recordings. You see in several parts of the world. On the upper left you see a recording in Italy, Valdieri in the Italian Alps. On the right you see our recording site in Borneo, Kinabalu Park, Sabah, Malaysia, which I already mentioned. And you might recognize that I'm standing there on a canopy walkway for researcher, which are much infrastructure, and we distributed microphones there and recorded soundscapes in the 90s, which I have digitized as mentioned, and a lot of which have already been uploaded to Bioacustica. You heard already about Bioacustica, and Ed Baker is here to answer questions about Bioacustica. So these soundscape recordings, of course, are distinct from the identifiable voucher recordings, but I'll talk about that later. And on the lower left you see my companion from the indigenous group of the Sequoia in Ecuador, who accompanied me during my recordings in lowland Amazonian Ecuador. And right below here you see our Panama Barra Colorado recording site. And as you can imagine, the soundscape recordings not only contain Orthoptera, but also contain birds and frogs, and you are all welcomed to reuse these recordings and send me information about the other animals which are there, moving towards an ATBI in acoustics, an all taxa bioacoustic inventory. On the other hand, of course, as orthopterists, we are interested in your recordings. from vertebrates, because most recordings from vertebrates, as for example frog soundscape recordings, of course contain Orthoptera. So these are some sonograms from the soundscapes I mentioned. In the upper left you have typical Orthoptera acridid songs, which are the short-horned grasshoppers, Grasshoppers sensus strictu, which you can hear outside in the campus, I suppose, in England, Orthoptera species, a typical broadband sound with a definite, well-defined temporal structure. On the upper right, you see the pouring examples, which we'll listen to in a moment, and on the lower, the Rio Aguarico, Amazonia, which with strong cricket bands, and the same for Barro Colorado Island, Panama. So all these broad bands here between 4 and 8 kilohertz are crickets singing on different frequency bands. So now let's at least listen to one example, and I selected the Borneo one. So you see this low-pitched sound here is what? Yeah, please send me the result for my personal statistics. It's a katydid. It's a tympanal phyllum katydid, which produces this atypical sound for a katydid. In South America, the katydids are all high-pitched, often reaching high in the ultrasound. But in Borneo, everything is the other way around. Frogs are flying, birds are not flying, and katydids are singing at a low frequency, and so on. And they are often confused with frogs, which are singing on a cricket-like high frequency. So that's already an indication that you need soundscapes to interpret. You need reference voucher files to interpret these recordings. So let's go to the extraction of snippets and filtering. So this is a Panama recording, and you see the distinct cricket bands, every species singing on its own carrier frequency, like an FM radio pool in a big city with 20 FM stations. And now let's filter and go to the 6 kilohertz station and extract a poor frequency singing cricket song simply by applying a filter. Very easy. Then you get a clear signal. Here the temporal domain, the oscillogram of the chirp, and the chirp is made out of pulses, and the pulses are repeated in a characteristic frequency. And ideally, you not only get this morphospecies, these ethospecies characterized by its song, but also you get the morphospecies. Now, we have a lot of cricket songs in our database, and what we did then is trying to do an automatic annotation. That means you don't need neural networks to do that. You only need the first steps like filtering, segmentation, and feature extraction. These signal characteristics, which I already mentioned. You extract them and you write them back into the database, and you get an annotated table with simple parameters. So the Orthoptera song repository of the DORSA project was used to annotate all 5,000 sound files automatically with sound parameters. Sound parameters were added to the database, and the annotated database is now searchable for simple features such as carrier frequency and pulse rate. So this allows us to find a certain recorded sound. Now, what is the vision? The vision is to use these recordings for species discovery, and particularly to record species presence and absence, because if you don't record a certain target species at its preferred calling time and in the preferred habitat, you can be quite sure that you don't find it. For species monitoring, of course, you can use it for species abundance, activity patterns, etc. And of course, you can use it to describe new species or to discover new species. Now, several caveats. Soundscapes for Orthoptera are distinct from most our recordings, which are restricted to the audible range. In contrast, Orthoptera have their ears in the front legs for the crickets and in the abdomen for the acridids, and they cover a much broader range. They cover infrasound with their cerci, which have hair receptors behind here, and they go high up into the ultrasound with their ears. So, likewise, their song can be in the ultrasound and you miss it. So these are two recordings from Greece, where he made recordings, and you see the tetragonids here calling Perzilimon between 15 and 30 kilohertz. And here you see below you see the typical grasshopper song, which you do catch with a normal sound recorder. So you only get a certain fraction of these songs, which might be enough, but not necessarily is enough to detect the species. And here you see in bold those species which have high frequency components, and you see the Rhacoclais werneri you probably won't catch with a normal recorder. So, the second caveat is the low number of species covered in our databases. You see, Europe is quite well covered with 30% of the known species with audio recordings, but the other areas are really not covered at all practically. So if you evaluate the Orthoptera with audio recordings in the Tierstimmenarchiv, Bioacoustica, Macaulay, and other sources queried via GBIF, e.g. iNaturalist and other observation databases, then you find out that only less than 20% of Orthoptera species have sound recordings at all. So the challenges are huge. There's a huge potential for Orthoptera monitoring and species discovery by reusing recordings from numerous soundscapes projects. So this is big data. But there's also a big data gap due to incomplete sound libraries. Orthoptera reference recordings only for less than 20%. Then we have the taxonomic impediment. Hitherto undescribed species require databasing of operational taxonomic units or ethospecies. Lack of annotation software, even for simple features. Lack of protocols for acknowledgement, traceability, metadata exchange, and metadata inheritance to sound snippets. So thank you for your attention, and thanks to the DORSA team, Klaus Gerhard Heller and Siegfried Ingrisch, to the Orthoptera Species File team, Maria Signano, Holger Braun, and my co-authors from the media. Thank you very much.